Hello everybody, thanks for joining to our new webinar. Today we will talk about DNA fragmentation, its relationship with oxidative stress, and what is the best way to handle with that problem in the IVF labs. So to start, I would like to clarify a term that is commonly confused, that is the DNA damage. It's important to know that DNA damage could be triggered by different processes, like could be defective packaging on the DNA during spermatogenesis, processes of cell death, and oxidative stress associated to pathological and environmental factors. Okay? This could involve chemical or physical modification on the structure of the DNA, including deletions, changes in methylation, incomplete packagings, base modifications, cross-linking, or strand breaks. Important to know, we are talking about sperm DNA fragmentation only when we are referring to single or double strand breaks. Okay, so the DNA fragmentation is only a type of the possibilities, the possible types of DNA damage assessed in a semen sample. There are different methods, different techniques for assessing DNA damage on ejaculate. For instance, the acridine orange test that is very useful for labeling specifically the single strand breaks. This is commonly used with fluorescence microscopy, but it fades so fast, and sometimes it's difficult to assess manually, okay, because the acridine orange is binding as a monomer when the DNA chains are intact, so getting a green coloration. But when the DNA is broken, it is bind as an aggregate and produce a shift on the coloration to yellow or orange. Therefore, evaluating the shift of the coloration of the sperm cells, you can quantify the level of single strand breaks on that sample. But it's not easy to do manually. This is why this fluorochrome is normally combined with the flow cytometer, because once you are incubating a semen sample with a denaturing solution, so the DNA that is damaged, that is broken, is very sensitive to the decondensation after this treatment. Therefore, it's easy that the acridine orange bind to that this breakage, and therefore you can assess accurately with the flow cytometry these coloration shiftings on the sperm sample. This is the conventional or the commonly known as Scsa method, in which we are analyzing the cells that are denatured by incubating with an acid and then stain it with acridine orange. It allows to calculate the DFI of single strand breaks. Okay. Also, there is possible to evaluate the level of maturity within the ejaculate. This is the reason why it's recommended to use aniline blue staining, in which there is a specific labeling for histones. Important to know that immature sperm cells contains high percentage of histones. This has not been replaced by protamines during maturation. Therefore, the compactation of the nucleus is not very good and involving some problems during fertilization. Therefore, you can do a specific labeling with aniline blue through these histones and evaluate the ratio between histones and protamines. The CMA3 staining, the chromamocin, it is also used for evaluating these non-well packaged DNA, this chromatin that is not fully compact, because this chromomer has a high affinity for the relaxed regions of the DNA, these that are not well compact, and therefore there is an indirect method for evaluating the protamine deficient DNA. Concerning the evaluation of problems on condensation, the toluidine blue staining could be used. Okay, in that case, the toluidine blue has a high affinity for binding to the phosphate residuals from this chromatin that is not well packed, is not well compact, and therefore could be indicative of the condensation, the level of condensation of that chromatin. Indirectly, it could be used as a measure of DNA breakage, because the DNA that is broken is not fully well compact, and therefore could be an indirect way just to assess this fragmentation. Okay. Also, it is very conventional for assessing this chromatin strand breaks, the use of the tunnel test. This is specific for labeling the nicks on the DNA. Single and double strand breaks is very specific, but it has a problem for telling on that way. That is only labeling these cells with a high level of damage that are pre-apoptotic. Therefore, all these sperm cells with a low or intermediate level of damage are normally dismissed. Concerning the sperm chromatin dispersion test, we will talk about this later, okay? But this is a method in which the semen sample is encapsulated in an agar gel and treated with a denaturing and a lysis solution. It is very useful for assessing the level of damage and quantify the level of single and double strand breaks. Okay, so this is very useful in the conventional laboratories. It's easy to reproduce. And finally, there is also a possibility to do a comet assay, a comet test, in which the semen sample is treated with a lysis solution and then is passed through an electrophoretic gel, in order that all these chromatin fragments are spread all over the gel, generating like a comet tail. And hence you can just quantify the level of damage or the level of DNA fragmentation that is inside the sperm cells. Here I have included a table that is summarizing what are the principles and the advantage and the disadvantage for each technique. Okay, it could be useful for having a look later. And just to clarify that for assessing DNA fragmentation, the most accurate ways to do that are the tunnel, the Scsa method, sperm chromatin dispersion or halo test, and the comet assay. These are the methods that are more specific for detecting the single or the double strand breaks. So, it's important to think in mind that the lifestyle factors or systemic factors, like could be the obesity, smoking, the frequency of ejaculation, maybe if there is any problem in the genital tract, if there is any infection, these are primary sources of oxidative stress. And this oxidative stress could involve alterations in the homeostasis of the testicular tissue, therefore inducing physiological and pathological problems on the ejaculates. It is commonly that under high levels of oxidative stress, sperm cells ejaculate have problems for capacitating, for undergoing the acrosome reaction. Normally acrosomes tend to react prematurely, have a lot of problems for hyperactivation, and for recognizing the oocyte. and therefore for ensuring the fertilization, the natural fertilization. On a second level, this oxidative stress could affect on the viability of the membranes and on the DNA structure, inducing problems of peroxidation, especially on the midpiece, on the heads, and involving the apoptosis when there are high level of damage on the sperm DNA chromatin. Okay, and all these issues could obviously have a very important effect on the success of the IVF treatments. So, in about of 25-30% of cases in which there is not a real identified cause origin of the male infertility, these cases are normally related with high percentage of DNA fragmentation. Commonly, the oxidative stress is its main contributor. In this paper from Dr. Agarwal, it is quantified the impact of the oxidative stress on the infertile couples. So, oxidative stress is affecting in more than 50 million couples all over the world. Okay, so it has a huge impact on fertility clinics, and it could involve lots of problems during embryo development. Okay, normally involving miscarriage during the first stages of embryo development. So, this is a very important parameter for being controlled in the laboratories. Actually, recent papers are evidencing that its prevalence on fertility clinics is very high. According to data provided by Dr. Esteves from Brazil, more than the 50% of patients that are assisting to a fertility center have high levels of DNA fragmentation. Okay? This result has been also observed in different clinics. So it's important to pay attention to the origin and what are the causes for this DNA, this high percentage of DNA fragmentation in our patients. This is why it's important to try to assess the origin of this fragmentation, and it is coming from the oxidative stress that is occurring in the male tract during sperm maturation. This is a very interesting picture, a scheme, in which Dr. Kenning has quantified the level of fragmentation on different stages of the male tract. And what is very important to know is that, very interesting, is that the level of fragmentation in testes is very low compared to that found in the ejaculates. So, what is happening here? There are so many publications that are revealing that is specifically the transit through the epididymis that is involving the release of a high level of reactive oxygen species that could affect on the motility or the membrane features on the ejaculates. This is why for so many clinics, actually they are applying a specific method for treating the oligospermic or severe oligospermic patients, in which they are doing the ICSIs or the IVFs from samples collected from the testes. Okay, so here in that paper you will find exact values. Just to summarize, the percentage of fragmented cells is much higher in testes than in ejaculates, and it has a notable impact on the different stages of the IVF treatment. In that case, there is the testicular ICSI, ejaculate ICSI. So we are looking that the success of the IVF technique, on the Scsa technique, is much better when you are recovering testicular sperm cells, because the effect of oxidative stress during the pass of the epididymis is very low. Also, it's important to control the time for ejaculations. When there are long abstinence periods, there is a high impact of the oxidative stress on a stored sample, and therefore this is diminishing the quality of the ejaculates. Therefore, it's important to reduce this abstinence period through 1-3 days, not more. Also, it's important to take in mind that depending on the method that you are using for assessing DNA fragmentation, the clinical cutoff will be a little different for each technique. So these are some examples in which there are compared the sensitivity and the cutoff values for different methods. So overall, the tunnel, the SCSA and the CD techniques show a cutoff, a sensitive cutoff close to 20%. But when you are using COMET, this cutoff, clinical cutoff, is much higher. Okay, so depending on the sensitivity of each technique, this cutoff should be adjusted to get a good interpretation of the results. So we have seen that DNA fragmentation is a very important parameter to assess, especially for the IVF clinics. And this is why in the last edition of the WHO guidelines, it has been incorporating or have highlighted several techniques for assessing directly or indirectly this parameter. So, as is proposed to be as extended examination of the ejaculate to assess the DNA fragmentation, but also as advanced examination to evaluate the effect of the oxidative stress in your patient and evaluate the structure of the chromatin on the ejaculate. So this is important. Why? Because when you are doing a conventional seminogram in which you are evaluating motility, concentration, morphology and vitality, you can get optimal results for all these parameters, but then when you are doing a treatment, there is no fertilization, or maybe you can find high percentage of embryo miscarriages. This is on that profile of samples in which DNA is especially recommended, also before any reproductive treatment, or maybe when there is an advanced paternal age. Again, the environment, all the homeostasis of the tissues are affecting on the levels of oxidative stress and on the probability to find some errors in the methylation, or also on the sensitivity of cells to be damaged. Therefore, paternal age should be also taken into consideration for evaluating the level of damage on the DNA. So, the most commonly used technique on the andrology or the IVF laboratories is the sperm chromatin dispersion test, because it's easy to reproduce. You don't need any specific device for assessing, and this principle is based in the encapsulation of a sperm sample on an agar gel that is denatured and then lysate. So when there is intact DNA, this is fully compact, it is well packaged, and is very sensitive to denaturing solution. And this will display big or medium halos once you break the membranes. Conversely, this DNA that is fragmented it is not sensitive to denaturation. And therefore, when you break the membranes, this will not react and will remain as a compact heads. So with that test, you can evaluate 5 different categories. Those sperm cells that display big halos and medium halos that are assumed as good quality sperm cells, but also when you find small halos, no halos, or degraded, should be assessed in order to quantify the bad quality sperm cells within the ejaculate. This is not easy to do manually because it's not possible to measure accurately the size of the halos. This is why the Scsa systems, the Scsa system, allows to perform an automatic and very fast classification of these sperm cells of this technique, and provide a very detailed information of the percentage of fragmentation, but also a detailed information about what is the percentage of sperms with big halos, medium halos, small halos, and degraded sperm cells. And you can show it, including these main results with graphs, with images in a very nice and fancy reports. Additionally to that test, MICROPTIC also provides from complementary tests, like could be the CAN-ROS, that allows to assess the overall reactive oxygen species effect on the ejaculate, or the concept kit that allows to do a sperm selection in which it will be dismiss this sperm cell with high level of DNA fragmentation. This is based on the annexing columns that is passed through a magneto, and therefore these cells that are damaged are attached to the walls of the magneto, okay? And the sperm cells that are not damaged will be swimming freely all over the tube, okay? If you want to know more information about that product, don't hesitate to contact us. So, once in the laboratory, we detect that there are lots of problems, high levels of fragmented DNA on our patients, what to do, what we can do. Okay, so here I have included a brief scheme of what are the most used techniques for minimizing this DNA fragmentation. So, commonly, the reproductive assisted clinics recommend using antioxidants before collecting new samples in order to minimize the effect of oxidative stress. Okay. Also, it's important to control the lifestyle of the patients: smoking, drinking, having some medicines. All these environmental factors could affect on the quality and on the levels of oxidative stress within the ejaculates. Therefore, should be controlled as much as possible. In some cases, it's necessary to do some surgeries. So here, normally there is when there are problems of varicocele that are normally released, are linked to high temperatures and high level of reactive oxygen species. The best way is just the surgery, okay? Or when there are infections, to treat with antibiotics. Also, in some cases, it's necessary to treat with hormones. And in the assisted reproductive labs, normally they are directly going to Scsa, okay? But what we can do to minimize these levels of DNA fragmentation on our labs? There are different methods, different ways to control that parameter. For instance, recommend a pretreatment with antioxidants before the donor releases the sample. Reduce the abstinence time period to 1 to 3 days. Decreasing the time between ejaculation and fertilization. Okay, you need to synchronize the process of sperm collection, analysis, and fertilization in the lab. Use ejaculate at room temperature. Important, if you are storing the samples at 37 degrees, the metabolism of sperm is 100% active. Therefore, there is a high probability to find to release a lot of reactive oxygen species all over the media and trigger the damage of membranes and release of more reactive oxygen species all over the ejaculate. So we need to control the temperature to minimize, to work at room temperature, to minimize the metabolism, reduce this activity, this motility on sperm cells, and reduce as much as possible the release of reactive oxygen species to the seminal plasma. Obviously, we need to remove it as soon as possible. We can do a sperm selective washing like swim ups or density gradient, remove all these source of reactive oxygen species from the sample and select the best part of the ejaculate. For doing the sperm selection, there are different ways to do that. For instance, there are the principle of MAX, of magnetic sorting, similarly than this kit provided by MICROPTIC. There is the option to do PIXI, IMSI, use microfluidic kits, density gradient, swim up, and combine abstinence with some washing methods. Here you will find the average relative decrease on the DNA fragmentation performed on each of these methods. Okay, so you can just have a look to this publication in order to evaluate what is the best way to reduce this DNA fragmentation in your routine, in your laboratory. Okay. Also, is important in cases of extreme oligospermias to consider a possibility to use for ICSIs the testicular sperm cells instead of the ejaculated, okay, because these are not so fragmented. And a very important point, control the sperm manipulation in the lab. I have added that publication that reveals the huge impact of these conventional methods applied in the laboratory on the release of reactive oxygen species on the sample. So centrifugation, high temperatures, changes on pHs, prolonged cultures, cryopreservation are affecting a lot on the homeostasis on the sperm sample and the oxidative stress induced by reactive oxygen species. Therefore, should be controlled as much as possible in your lab. And one frequently asked question there is, okay, I want to assess DNA fragmentation in my sample, but I have a lot of things to do in my lab. What is the best moment to do that test? Important to know, if you are storing the sample for long periods, the level of oxidative stress will trigger an increase on the DNA fragmentation on your sample. Therefore, storing conditions should be controlled as much as possible. So, for instance, when you are assessing for DNA fragmentation 4 hours after collection and this sample has been preserved at room temperature, the percentage of fragmentation is practically 2 times the original ones. Okay, so here there is a huge impact on the storing conditions and it could affect on the interpretation of results. Okay, when stored at room temperature for 24 hours, you can see that the DNA fragmentation is passing from 15 to close to 50%. Okay, this is a huge change in the percentages. So it's important to control what is the best way to assess it as soon as possible, and you can preserve the sample on the fridge at 4-6 degrees for some hours, but not more than 4 hours. Okay, so this test should be ideally performed on the same day. So that's all for today. Thank you so much for your patience, and now is your time for asking questions. If any doubt, anything, once you see this webinar, you can also contact me at that mail, nuria@micropticsl.com.